Genomics. What my genes say about me. Let me introduce Sylvia and Olaf. Sylvia loves dark chocolate, while Olaf is not very fond of it. Today they are going to do a test to find out why. These paper strips are impregnated with theoria, a very bitter substance. When Sylvia and Olaf taste them, Sylvia doesn't notice anything, while Olaf detects a horrible bitter taste. Now the doctor is taking a sample of their saliva. The doctor will analyse a tiny molecule contained in the sample. It is called DNA, and it has a lot to tell. The doctor finds that there are small differences between Sylvia's and Olaf's DNA. These may explain, at least in part, the difference in their sensitivity to bitterness. The test you have seen is just a simulation. These kinds of analysis must be done under medical supervision for both ethical and scientific reasons. However, it is true that certain features of the DNA influence taste. If you are curious to understand why, let's explain how DNA works. DNA means deoxyribonucleic acid. This molecule is packed inside every single cell of our body. If we stretched it, it would look like a very long text, written in only 4 letters, the bases. A for adenine, G for guanine, T for thymine, and C for cytosine. The bases combine to form sequences, something like words in a text. Some of these sequences are called genes. They contain the basic instructions of life. The cell has powerful machinery that transcribes the DNA of the genes into another molecule called RNA. It then translates the RNA into proteins. Proteins perform essential functions in the organism, from building organs to distributing energy. The conversion of genes into proteins is called gene expression. Let's go back to Sylvia and Olaf. The result of the differences in some of their genes means that they have different proteins that are bitter receptors. This results in different sensitivity to bitter tastes. It is not just one gene that plays a role. In fact, many genes and even DNA sequences that are not genes interact in the expression process. Moreover, things like education, culture, environment and social class end up influencing expression. Now, consider that genes do not influence only taste, but all the features of our organism, whether we are tall or short, the colour of our hair and eyes, even our temperament may have a genetic component. If certain genes don't work well, the organism can become ill. Scientists can also modify genes to improve the features of plants and foods. For all these reasons, scientists are striving to improve the technique for reading DNA, which is called DNA sequencing. Genomics is the science that does this and interprets the meaning of the code. Physicians are starting to use genomics to assess the predisposition of patients to certain diseases, or how well or badly an illness could progress once it has appeared. Moreover, a treatment that works for your genetic profile may not work or may even be toxic for mine. The pharmaceutical industry is working on drugs for specific gene profiles. DNA tests are also used for drug trials. A decade ago, the first whole human genome was sequenced. Since then, the rate of sequencing has become 50,000 times faster, and the cost of sequencing has become 10,000 times cheaper. It now takes less than 1 month and a few thousand euros to obtain the genome of an individual, and these figures will decrease rapidly. In the meantime, thousands of other organisms have been sequenced. Maybe one day you may get a blood, urine or saliva test and receive all your DNA sequence in a USB drive. The doctor will plug it into her computer and find your predisposition to certain diseases, prescribe medicines specific to your profile, and design the most suitable diet for your genes. This is the promise of personalised genomics. We are just at the beginning, but scientists are moving fast in that direction.